So it's a great honor to present at this seminar. Thank you for the organizers and for everyone listening. This is our most recent paper, Predicting Functional Constraints Across Evolutionary Timescales with Phylogeny-Informed Genomic Language Models. And this is something that was released on bioRxiv on September. And also to mention that Chengxiong Ye, who is a co-first author, I think he might join and also kind of offer some perspectives later. So first, an introduction. The main problem that we're trying to solve here is understanding genetic variation. For example, given a mutation from G to A, we want to know what is the effect of this. Is it benign, neutral, pathogenic, deleterious, beneficial, adaptive? And in a very small proportion of the cases, we know that this causes hemophilia B. But this is literally, we only know like a 1,000 or so, couple in the order of thousands of variants that we actually know exactly what they do. And the general picture of the problem is when we consider the full map from genotype to phenotype. The genotype is a collection of, let's say, the DNA, could be in an individual, for example, with all the mutations. And this determines the expression of proteins across different tissues, different conditions. And this then determines the phenotype, the high-level phenotypes such as height or disease resistance, for example. And then natural selection closes the loop by selecting which genotypes continue onto the next generation. Now, we cannot really model this entire process. We only have limited information, data so far. And for example, functional genomics supervised models predict the gene expression from the genotype, often even just using a single reference genome, and typically not predicting the protein expression, but just the RNA-seq levels. On the other hand, there's self-supervised models that predict the genotype from itself. And the reason this makes any sense at all is that there is a closed loop that because of natural selection, so the genotype itself contains somehow implicitly a lot of information about all the steps in this system. And more precisely, these self-supervised models that we refer to as genomic language models. The main idea is similar to ideas from natural language processing, where a certain parts of the sequence are masked, and you want to predict them from the context. So this you use, let's say, something like a transformer or a neural network. You generate a contextual representation. You aggregate information from around the mask site, and then finally, there's a language modeling head that predicts the hidden nucleotide. And what you can do with this is, for example, score variants. Typically, the ones that have low probability are associated with disease. This is kind of an empirical observation. And this is what, in general, we call functional constraints, because it's not just disease, could be any fitness effects. And then we can also sample from this distribution to generate new sequences. And another application is transfer learning, where we kind of adapt with a new kind of layer on top. For example, if you have labeled data of gene annotation or gene expression prediction, and you fine-tune the model. But this talk is only about functional constraint prediction. So one of the key ideas is homology. So the similarity due to shared ancestry. This can be seen in the anatomy. For example, different parts of the body that have basically come from the same origin and have a similar function in many cases and similar structure. And more importantly to this talk is sequence homology. Let's say you can look at the histone protein among different species. And basically the assumption is that this protein basically has the same function, or roughly a lot of the same function and structure will be. And you can see information on which parts are conserved versus non-conserved, for example. That is really useful information. And the general message is that, homologs always provide clues. They're not exactly the same as the system that you're studying in human, but it's always some information you can get from looking at homologs. And this is a critical component of AlphaFold, which has basically, starting with an input sequence, it does a search for similar, basically homologs, and basically represent them as a MSA or multiple sequence alignment. And this actually is also really common in LLMs as well, the idea of retrieval augmented generation. So when you start with a query, instead of just generating a response directly, you first basically do a search for similar documents in a database. And then you add them as context into the inputs for the LLM, again, to generate the response. And this has several interesting features about this. The idea is that you can decompose knowledge. So you can, on one hand, the LLM can handle like language, like syntax, the grammar, the reasoning, and the database can handle factual knowledge. So this generally leads to more accurate results. You can like control the information to have like to keep it up to date, and you can use like smaller, faster, and cheaper LLM. But there's a complexity to maintain the database, and it's not always clear that you can decompose the knowledge into, let's say, procedural and factual. But the question basically is also very present today, like in biology, because homology is still captured implicitly in model weights in single sequence language models. But the idea of retrieval augmented generation is about making the homologs explicitly available to the model. And maybe if we think about in terms of, let's say, the bitter lesson, suggests that we should just do as few assumptions as possible and just, let's say, scale up models. However, so far, the picture is not that clear, or maybe it's actually quite clear that retrieval is really important, especially in proteins. Has been shown, for example, for AlphaFold and protein structure prediction. It outperforms ESM fold that is based on a alignment-free model that is much larger, 15 billion parameters. And furthermore, the MSA is really important. When you ablate it, the performance drops down significantly. And also, people have observed that for variant defect prediction, sometimes there's a point where further scaling basically reduces in worse performance. And this is one of the ways people understand this is that the model is basically learning phylogenetic biases instead of functional constraints, so somehow overfitting. And what about DNA? Well, maybe one of the greatest, largest experiments with scaling is the EVO2 model, up to 40 billion parameters. And we did some independent benchmarking in TradeGym for regulatory variants. And we found that it actually scales with scale from 1 billion, 7 billion to 40 billion. It does improve the performance in many cases. This is, for example, in promoters. However, it doesn't beat CAD. So CAD is a model that is just a logistic regression model trained on really, really powerful features. And one of the main feature is the conservation in the MSA. So in some way, it still is not clear that scaling will really help for, let's say, human genetics. And furthermore, we found that the EVO2 struggles the most with distant enhancers. Basically, as you scale, it is really, really far away from the performance of the CAD baseline. And we believe, we discussed this in the TradeGym paper, but it is because enhancers are just very sparse in intergenic regions. And it's more of the challenge is kind of like a data curation. So, with this, it is basically we started working on alignment-based genomic language models. So basically, instead of just predicting a sequence from the context around it, let's say in the genome, you can also use the context across species, across evolution. And this is our work, GPN-MSA, and it actually does, it brought non-trivial improvements over previous models. But there was something that we wanted to improve in the next project. One, it is about the architecture, that GPN-MSA was just kind of fusing all the nucleotides into a single token, all the nucleotides in a single column. So it's not very flexible. The loss was only computed on a single species, on the human, so it's losing training data. The model is unaware of evolutionary relationships between the different species, and it was only trained on a fixed alignment, the vertebrates. And I mean, we have many reasons to believe that may not be optimal. So now I want to talk about our most recent model, GPN-Star. So it has two ingredients. One is the whole genome alignment. So for example, if you look at the ACE2 gene, here are the exons, let's say the little blue bars, and you can see that they are conserved across vertebrates. But if you look at the introns, they are more like they are conserved or aligned only to mammals. And maybe some parts, let's say if there is some parts are only aligned to primates even. So there's all this kind of complex alignment. And then we have the phylogenetic tree of all the species. And so GPN-Star stands for Genomic Pretrained Network with species tree and alignment representation. And one of the characteristics is that it separates into target and source sequences. Basically, instead of just predicting only the human sequence, basically there's a random selection of species that are used for prediction and some that are used as context. Another thing to highlight is the idea of clustering sequences into clades based on a hyperparameter. And this has many important reasons why we do this. One is to save compute. Another one is to somehow balance so that if a clade has a lot of sequences, it will basically only be collapsed into a representative one. And then control information flow. This is something that we had observed that, for example, if you're trying to predict in human and you give it the chimpanzee sequence as context, we somehow believe that the model is just kind of copying. It is too easy of a task, and it leads in worse performance. So it's really important to put some kind of boundary between the query and basically the target and the sources. And the way we use another way that we use the evolutionary distance is by adding basically a bias term in the attention, basically. So it is still a flexible function. It's not that we explicitly add the evolutionary distance, but this is one parametrization that is really easy to work, and this has allowed us to basically we're having like one hyperparameter that is the kind of a threshold of evolutionary distance, and that has been working pretty well across different alignments with some small variations. So we trained on 3 evolutionary timescales that are relevant for human genetics. We have previously we had the vertebrates, but now we only have on mammals and only on primates. And why is this important? Well, I mean, recent papers, for example, from the Sunomia consortium, have shown that certain, for example, say for complex traits, like primate-specific constraint is really important. And this is something that we explore in the paper as well. And we will call this abbreviate like V, M, and P. So we found that GPN-Star is state-of-the-art disease variant predictor. So if we look at, let's say, coding variants, one very standard database is CleanBar. And here we're highlighting the best of the three models. So the vertebrate model, not surprisingly, is the best here. So let's say for things like proteins that are kind of very well conserved, using a longer evolutionary timescale is very helpful. And on the other hand, for the non-coding variants, we found that the mammal model is working better. And here again, we compare against, for example, models such as EVO2 and nucleotide transformer that are really large genomic language models, and also against like AlphaGenome, for example, and other sequence to function models. So then we look at causal variants from GWAS. So this is not necessarily disease causing, it's any GWAS trait. And we use fine mapping results. So where we know that a variant has a high probability of being causal versus low probability of being causal. And we find here again that the model, in this case, the mammal model is doing the best for both coding and non-coding. And if you look at, for example, on the non-coding, you could say that AlphaGenome is really close. And it's really interesting here to highlight because these models are kind of pretty much orthogonal in the data that they're using. So it looks like you could get like basically, if you somehow, the idea of combining the two approaches is very natural, and I think it's for sure being more emerging recently, and like a central challenge, I think, for the future. Also, if you look at promoter variants, it's interesting because there are kind of models that are specific to this. So we also highlighted along two axes. It's kind of the GWAS fine mapped, so it's kind of on complex traits on the x-axis and Mendelian disease on the y-axis. And generally, you can see that the sequence to function models are better on the complex traits, whereas if you look at conservation models such as CAD and GPN-MSA, we're doing better on the Mendelian disease. But the GPN-Star is basically doing great on both. Next, we looked at complex trait heritability enrichment. So this is a great framework because it allows you to use the signal across all SNPs in the genome, like 10 million variants, rather than just using fine maps that are only like a couple thousand. And the main idea is that basically under a polygenic model, you have the heritability, that is the proportion of the phenotypic variance explained by genotypic variants. And then when you look at specific annotation can be, for example, it could be coding variants, could be enhancers, or more in our case, we look at the top conserved sites according to a model. It has a certain, let's say the top M SNPs. So then what we do is calculate the heritability enrichment is kind of the proportion of heritability associated with that annotation C divided by the proportion of SNPs of that annotation. And we did this analysis. Here is showing like the meta-analysis across 106 GWAS traits. And again, using all SNPs in the genome. Even the good idea about this framework, the good thing about this framework is that it doesn't require nor fine mapping, not even genome-wide significance. So it can really use all the signal, even if it is like below significance threshold. And we use here the top 0.1% variants as a notation. So this is something roughly like 10,000 variants. It's kind of, we ask each of the different models which are the top 10,000 variants, let's say candidates to be functional variants. And what we found is that the most enriched is basically the top variants according to the primate GPN-Star model. And this is seen both in the heritability enrichment and also in the conditional effect. So many people think that conditional effect is more interesting because it's conditional in what we already know. We have like 100 or so baseline annotations, such as if it's coding region, if it's enhancer, things like that. So it's good that on top of all the knowledge that we already have, how much signal you add. And it's also worth noting that the previous state of the art was fast-cons, that is a conservation on primates. So this is something from, again, from the last 2 years or so that was shown. And it was great that we somehow also see the same pattern, that primate-specific constraint is the best. And when we separate into coding and non-coding, we find that the main advances are on the non-coding region. And then we looked at polygenicity. If we zoom into specific traits, there's this estimation of effective polygenicity, that is roughly how many independent variants are contributing to a trait. We look at the one end of the spectrum, LDL cholesterol is expected to have around 89 independent signals, whereas schizophrenia has over 13,000 independent variants contributing to it. And on the y-axis here, we're plotting the difference in enrichment between primates and mammals. So where we see basically below 0 means that the mammals have a higher enrichment. So the mammal GPN-Star model. And above the 0 is where the primate model is doing better. And we found a moderate correlation. So the more polygenic the trait, the more that the primate model is enriched compared to the mammalian model. So this is something that, I think it's pretty mysterious and worth investigating further. Another thing that we did is think about tissue specificity. Many of we know that certain traits are acting, the mechanism of how a disease develops is kind of particular to a specific tissue or set of tissues. But GPN-Star and in general, all conservation scores, they have no idea of the concept of a tissue. Basically, natural selection, if you see basically the conservation pattern, it's somehow aggregating all the different tissues in whatever non-linear way that is kind of a black box. We have no idea which tissues are involved just by looking at conservation. So we did this approach that is adapted from LDSE-seg, that is basically you look at the tissue-specific genes based on an RNA-seq data, and then you find the top variants basically only around the differential expressed genes. It's kind of very simple experiment, but we did see cool results. For example, for brain traits, if we basically take the GPN-Star model and only look at the top variants within brain-specific genes, you have an increased enrichment. Similarly, for example, for blood immune. And we also see something similar with Informer, where it's not exactly the same way of adding tissue specificity because the model is already tissue specific. It's about how you aggregate the different tracks. And again, in specific traits like schizophrenia, you see a big increase in enrichment when you look only at that specific around the genes that are brain specific. And finally, to summarize our findings, we basically have two axes of these are all different kind of benchmarks or variants or variant effect prediction problems. Basically, you have the coding, non-coding, and we have from low allele frequency to high allele frequency, or from really strong large effect sizes to more subtle effect sizes. And maybe starting with the more popular benchmarks such as ClinVar, coding variants. There we find that the vertebrate model is the best. On the other hand, if you look at kind of Mendelian disease, but non-coding variants, the mammal model is doing better. And on the other hand, we go to the really subtle effect sizes of non-coding GWAS variants, the primate model is doing the best. You might point that there is some discrepancy between the fine mapping and the stratified LD score regression. And I think that is a pretty large puzzle that I have no idea. I suspect that the LD score regression variants are still somehow smaller effect sizes than the variants that pass the fine mapping, but I don't know if that is actually true. And some other thing that we found, for example, we did this kind of UMAP in the original GPN paper, where you basically look at the embeddings of different genomic windows, and there was clustering by genomic region. And we found something similar, even with when using an alignment. So the model is still learning a representation somehow of the context. Although if you look at the non-conserved sites, they kind of, basically they're all basically lumping together all the regions. So this is something also to understand what that means. And another interpretation that I really like is the nuclear dependency analysis. So people always look at this and think that this is kind of an attention map, but this is more general. It's a black box approach. You just modify the input, and that is the DNA sequence, and you see how the probabilities that is the output of the model changes at other positions. And in this case of this promoter, we have data about known transcription factor binding sites, basically these 4 that are marked here. And we see that the nuclear dependency suggests that the model is kind of predicting that these nucleotides within a TFBS are interdependent, as we would expect. And furthermore, it also predicts interdependencies between, let's say, pairs of transcription factor binding sites. And in this case, they're actually known to physically interact. So this is basically like I think this makes pretty clear how this model is an improvement over phylop, kind of the most one of the previous conservation scores that only looks at a specific alignment column. GPN-Star, on the other hand, is clearly looking at the context in like non-trivial ways. Not even within not also both within a transcription factor binding site and also looking at nearby transcription factor binding sites. And another really cool analysis that I think it could be its own paper, because this is a pretty important conceptually, that when we look at variation, let's say variants, there are 2 processes that are basically generating this variation. There's first a mutation that is, it is heterogeneous. There's a different mutation rates across different parts of the genome. And then there's the natural selection that basically selects which variants will cause disease or not. That is kind of what we care about. So to control this basic mutation rate is a confounder. We want to control for that. So what we did is kind of generate like a neutral model that is basically finding neutral sites in ancestral repeats that are not conserved. And we calculated the average model scores for each 5-mer context. We know that the 5-mer is important for the mutation rates. So we calculate this statistic. And then we somehow adjust the low likelihood ratio, that is kind of the main model score, so subtracting kind of the score from the neutral model. And this basically what we find is that when we go from the raw log likelihood ratio to the calibrated one, it basically goes left in the sense that it's less correlated with the mutation rate. And we found that it's more correlated with functional constraint as estimated by the gnocchi model. And furthermore, if we look at some of the benchmarks, we basically see kind of 3 improvements in model performance by doing this correction. And yeah, I think like we will suspect that this probably applies to most models out there. It's not just for GPN-Star. And lastly, I wanted to highlight that we basically found all the alignments that were on the UCSC genome browser, basically. It was very easy to process the data in that case and just run the model with really just tuning a single hyperparameter that it almost works pretty out of the box without much tuning. That is kind of the phylogenetic distance that you use for defining clades. And the model is basically GPN-Star. Basically, that's better than phylopy and fastcons across the board. So, conclusion number one, GPN-Star is a state-of-the-art functional constraint predictor. So again, functional constraints, you might hear it called by many names. It could be the fitness, the deleteriousness. That's also related to pathogenicity, although pathogenicity could be causing disease. And in general, there are many effects on, let's say, fitness that are not through disease. And also, there's the concept of conservation. I see it as an estimator of functional constraints. Let's say if you look at the lines and you look at the alignment column and you look how conservative it is, that's one estimator. But GPN-Star, I guess we consider it a functional constraint predictor that is more general. And it does outperform fastcons and phylopy. These models have really stood the test of time. We do think that GPN-Star has improvements pretty much across all the systems that we studied. And it also performs the really large 40 billion parameter EVO2. But this is in the human genome. I still want to highlight that EVO2 is still an amazing resource for the majority of species that don't have an alignment. So I do think the idea of like alignment-free models is very useful in practice, especially for non-model organisms. And then maybe these two approaches of using functional genomics supervision versus self-supervised models that are functional constraints. So the way I see it is that today in 2025, specifically when we talk about trait-associated variants, so the models such as GPN-Star are doing better than the models that predict functional genomics, such as AlphaGenome. And this is, in general, you could say that if you have no selection at all, like the self-supervised models won't have a clue about the variant being having a functional impact. So in that case, it's probably that's one reason why we still want to understand functional genomics models, and like another reason is interpretability, like understanding the mechanism, how a variant causes a disease through which changing expression or epigenetic state in a certain tissue. All that is really important in practice if we want to develop, for example, therapies. And lastly, that these modeling approaches are not competitors. They are really complementary. And yeah, I think this is, I mean, there has been great work starting to emerge. For example, NONA on like basically multimodal modeling. I think there are many flavors. Maybe it could be like pre-training and then fine-tuning for gene expression, or multimodality, or even just kind of aggregating the scores at the end. That, in my experience, we have seen that actually improves prediction of, let's say, fine map variants, for example. And then that evolutionary timescale is key. The more polygenic the trait, the more the primate-specific model shines. And I think, I think our results that ranging from coding to non-coding and rare to common variants, I think they do highlight how different all these variant effect prediction problems are. And yeah, to try to really think specific about which task you're trying to solve, because they're really quite different. And finally, wanted to talk about MSAs. Are they necessary? So MSAs, they were originally motivated for proteins. And like we know that when you get further away from proteins, if you look at transcription factor binding sites, for example, like they can keep their function often, even if you rotate them or like change their order or spacing. And it really seems like MSAs are not optimal to represent such grammar. So perhaps basically the most surprising result of our kind of research program has been that these transformers operating on MSAs are still state-of-the-art across the entire genome, not just proteins. But if I look forward in the future, I predict that basically still like the idea of homolog retrieval will still be there, but the MSA might not be there anymore. And if you look at patterns, for example, in proteins, several models have shown that you can just concatenate the sequences, basically with a little spacer in between, and just the transformer will handle that and figure out where to look. Like you don't have to really align them. And similarly, you don't need to do sequence alignment to retrieve like the sequences. You can just do a vector similarity search. So I think probably system of the future will look more like this. And with that, I want to acknowledge all the people who made this work possible, starting with Cheng Xiong, the co-first author of this work, and Yun, who has been, our advisors supporting all this line of work for a couple of years. And I'll leave with that. Thank you very much for your attention.